Everyone else to please stay on mute.
Yes.
The front one is you can shut it off.
There we go.
Yeah.
Is that better?
Yeah. All right. Might be able to tweak this a little bit.
Great, touch it.
Can you sit down here?
Sam?
I should probably unmute myself. That'd probably be pretty helpful, wouldn't it? There we go. All right. All good? Look all good on your guys' end?
Yeah, it looks good.
All right. Right on. Well, I'm ready whenever you guys are. Give me the go ahead and I'll zoom. Okay. Awesome. Right on. Well, thank you, everyone. I appreciate everyone being here and letting me come and, well, not, I guess, virtually come down and present. I'm up here in Newport, Vermont, for those who are unfamiliar. If you got on, eventually got on Highway 91 and took it north, which kind of rides the border of New Hampshire and Vermont, and you went north until you got to the exit before Canada, you would find me and a small town called Newport. So that's where I am. I'm a high school science teacher, and I'm also the current president and longtime member of the nonprofit organization Trout Power. So here to kind of tell you a little bit about what we do as an organization. I've done a number of these presentations for Trout Unlimited chapters all over the Northeast. And I think the one commonality that every chapter that I present to has is the fact that, you know, we're in a region that is the home area of the brook trout. And that's what our organization really focuses our work on. We've kind of established, or working to establish, pardon me, a kind of a unique niche within the trout world, doing some genetic work and looking at some of the biodiversity of these native trout. So basically going to run you through kind of how we do that, you know, what we've done previously, what we're currently doing, and what's on our horizon. So we'll go through this, and obviously would welcome any questions when we get to the end. And like I said, I appreciate people being here and following along. So first off, just kind of starting with the generic here, you know, what is Trout Power? When I say that we're a part of this organization, we are a 501c3 nonprofit organization. We were established as that, you know, with that 501c3 designation in 2017. And you can see our mission, you know, statement on the screen. And really what we look to do is, you know, we're trying to go out and find and identify native brook trout, genetically unique brook trout, which within New York, the DC's kind of termed these heritage strain brook trout populations. And then once we, you know, find them and identify them, you know, try to advocate for them, monitor them, which I'll talk about how we do that. But the unique thing is kind of found within the first 5 words of our mission statement, which is that, you know, we are a completely volunteer-based organization. So we are really enlisting the power of anglers to really help us do this work, which I think is amongst a few things about us, one of the most unique and one of the most enjoyable things. So what we kind of consider ourselves is that we are volunteer anglers, but really what we are is citizen scientists. You know, citizen science it's a great way to do science. Like I mentioned, I'm a high school science teacher, so, you know, I try to encourage my students to become citizen scientists. And, you know, it's kind of cool that even if you have a different job title, you know, everyone and anyone can become a citizen scientist. So with that work, what we do is we have our own solo projects that we do. We've worked with other groups such as other Trout Unlimited chapters. We've worked with state level organizations such as the Department of Environmental Conservation. And then especially with our genetic work, we've worked with a variety of universities, currently working on different projects with Michigan State, University of Albany, University of Central Florida, as well as Cornell. So we kind of have a number of different hats that we wear. And like I said, I'll tell you about each one of those. But kind of starting with, like I said, with the uniqueness of citizen science, in case those terms, you know, together aren't familiar, this is an image from a event that we had, I believe, in 2018. And within that image, we have people who had been repeat longtime members of Trout Power and come to a number of events. We have people who have never been to one and just heard about what we were doing and thought they'd come and join us and see what we're all about. But everyone in that picture, the one thing they have in common is for that weekend in time in May of, I believe, 2018, we were all citizen scientists. So the idea behind citizen scientists is this is scientific research that's done either in part or completely by volunteer, non-professional scientists. So all of the field work, all of the data collection, if you will, is done by anglers using fly rods and barbless hooks. And then we send it over to the universities who really take they do the real hard science, and then send it back to us, and we get to check out what what we collected and what they helped us to identify, which is always a really, really exciting process. So thinking about this, again, one of the great things about citizen science is that, like I mentioned, we're all volunteers. So when, we receive calls from whether it's Trout Unlimited chapters or, the DEC, for example, especially with the DEC, half the time they're calling us saying, we have something we're trying to work on, or we have an idea, but we are overworked, underfunded, and understaffed. And so an organization that relies on passionate volunteers is, it's a nice resource and a nice partner for some of those groups to have. We have events, as you saw in that previous picture, where we'll have 25, 30 plus people join. So we're able to cover ground. We've had weekends where we've covered a really large, expansive area, and which is, really fantastic for our research. It's also a great way to connect the public to a cause. We've had people who have come to these events and come back for every one since then, and explored areas that they've never explored before. Again, all while wearing that citizen scientist hat, which is cool. And the last one, and there's my generic high school science teacher comment, where it's fun, and it really is, because you're fishing in beautiful areas for beautiful fish that may or may not be there. But I think the mystery of what you're looking for only adds to the enjoyment. So that's one thing that we really pride ourselves on, that again, we have no paid members of our organization. We're all volunteers. Yet through our volunteer efforts, we've been able to have some successes, which I'll gladly share with you all. So in terms of what we've done so far, what we've done since 2017, when we really established ourselves as a nonprofit, these are kind of the three, when I think about it, kind of the three major things that we've done, or the three categories, if you will, which I'll detail each one. But we've done what we call presence, non-presence studies, usually in conjunction with the DC. What we really have spent a lot of our time is doing that genetic analysis, searching for unique strains, looking for the biodiversity within our range, which is primarily the Adirondack Park. We've had one or two missions that have taken us a little bit west, kind of between the Adirondack Park Blue Line and Lake Ontario, the Tug Hill region of New York. But primarily, we really focus right now on the Adirondacks. And then as kind of a result of some of what we've identified in that genetic analysis is that we've also begun a couple long-term monitoring studies. And I'll talk about what those are, how we do those, and kind of the why, behind those long-term studies that we're currently doing. So first thing, like I mentioned, is that we have taken part in a couple presence, non-presence studies, usually in conjunction with the DEC. So the situation here is that we get a call from the DEC saying there's areas that they would like to know more about. Specifically, are there brook trout there? Are there fish there? And where are they? And again, the common saying with them when we talk to them is that they don't have the time, they don't have the manpower, and usually they don't have the funding or the money. So we get, assigned areas, and we go, and what we do is we go and fish those designated streams to see if there's wild fish that are present. We log everything that we catch and where, not just brook trout, but anything we catch, we log it. We mark any impassable barriers. So those impassable barriers are defined by the DEC, really related to the species. So for example, for a brook trout, if there's from the surface of your stream upwards, if there's a break of at least 18 inches, so that could be a waterfall, some sort of a culvert or something of that nature, anything over that designation, we mark on these maps as being an impassable barrier. Any other interesting landmarks that we find, and then we send all that information back to the DEC. So we're kind of the we're kind of the first ones to go in. One of our members and board members, Tim Bonaparte, says we're kind of like the black ops in a way, where they obviously a lot less dramatic and a lot less on the line, certainly, but we kind of go in and check out these areas, and then we report back to the state, and then they can send teams into certain areas that they have interest in, or if we go to a stream and we don't find anything, they can send a team into electroshock and confirm the lack of presence of fish within that stream. And later on, I'll talk about the shifts that we've had recently in New York in terms of stream management, because that gives a little bit more layering to these presence, non-presence studies and why we've done them. So that's kind of the basic run through. We've done a few of these. The first one that we did was in May of 2018, the year after we were founded. There's a wilderness area in the southern Adirondacks. If you went kind of north of Utica up towards the Speculator area, there's a big wilderness area called the Silver Lakes Wilderness. Remarkably expansive, a really beautiful area. Surprisingly, a lot of moose and bear sign for the southern Adirondacks, which was really, really cool. However, there's one road in and there's a set of campsites, and other than that, there's one trail system. There's really not a lot there. So we were sent the map that you see on the right side of the screen. And if you look at that map, I'm not sure how clear it is on the screen that you're looking at, but you can see that a lot of these streams are numbered. And every stream that was numbered was a stream that was assigned to us to go in and sample. So over the course of 2 weekends, spring of 2018 and spring of 2019, we were able to cover and fish all of those streams. So again, the power of citizen science and having, a large cohort of, motivated and passionate volunteers, we covered a lot of ground. And we found some streams that were chock full of little wonderful brook trout. And then we also found some streams that make a nice swimming hole and probably not much more because there weren't many fish in them. But again, the mystery is always kind of the part of the enjoyment of these experiences. So we've done a few others, more in small scale in terms of presence, non-presence, and we're currently working on a project to work with DC, as well as at least a chapter 2 of Trout Unlimited, doing some of this presence, non-presence studies in conjunction with looking at culverts and stream crossings, which is really exciting. So using this, kind of in a larger scope. So we're developing that now and hoping to get that rolling in the next, year or two. We'll see. So that's an example of one of these presence, non-presence studies that we've taken part in. So that's kind of our first major category. Second major category is what we really call our bread and butter, which is going out, catching fish, and then doing some genetic analysis on them to see, what do we have here? We're fortunate that we live in a region with wild native brook trout. In saying that, the DC stocks a tremendous amount of fish within our state. Since 1880, the DC has put millions of pounds of fish into our waterways, which, I've certainly benefited from that. I'm sure probably all of us in some way or form have benefited from catching stock trout. I know for me, when I was a young kid, 8 years old, trying to learn how to fly fish, it was a great confidence boost. When you see that truck pull away, you'd be like, oh, let's go catch him. So certainly have benefited from stock fishing, but there kind of remains that romance, especially in a place as remote as the Adirondacks, that, there are areas where, those stock fish haven't been put, and maybe those stock fish haven't reached. And so we can see that those fish are true native wild fish. So what I'll do is I'll kind of explain how we do this, what our process is in terms of before we go to a site, when we get to the site, and then afterwards as well. So what you could say is our goal is always to find and learn about fish. I guess kind of the dream goal, if you will, is to go and see if we can find some of what we call genetically unique strains of brook trout. So these are strains that have one of two originations. They are either a population of brook trout that have been in that location since the recession of, the last glacial ice age over 10,000 years ago, or they could be a population that was stocked privately, not by the state, but privately, and then has been able to take and remain over time. There's really no way to honestly say which one of the two there is. other than looking at private stock and records, which are pretty well documented within the Adirondack Park. So we always cross-reference those as well. However, if we do find, which we have found some of these strains that are, that are fish that have been there for a very long time, for thousands of years. The New York State, there's a term that's called the heritage strain brook trout. That's a term that was kind of coined by the DC with the release of a management plan in the late 1980s. But what it really means, these heritage strains, is those are fish that have unique genetics from other populations of brook trout and haven't been or have barely been influenced by the genetics of stocked fish. So that's kind of our, if there's a goal that we have when we go out, is we're always hoping, boy, maybe we're going to find another one of these strains, which again just shows the amazing biodiversity that we still have even after almost 150 years of stocking within the state of New York. Over on the left side, this is just an image that's from a place called the Wild Center, which is up in Tupper Lake, up in the north central Adirondacks. And this is an exhibit that's completely about heritage strain brook trout. You can see the title that's called the Adirondack 8. And I always smile when I look at that sign, because this sign in this exhibit was built before Trout Power was incepted, and now we've been able to give great evidence that the Adirondack 8 is actually the Adirondack 14, 15 plus, which is pretty exciting. So how we actually do this, when we choose a location to go to, what we do is that, first off, we reach out frequently to DC. We have a great partnership and a great working relationship with them. And we'll ask them just for any historical stocking records. If we get an idea, if we locate a place and we feel pretty good that it's probably a place that hasn't been stocked and we know it's not currently being stocked, we can check historical stocking records to confirm that. And then, we do our due diligence to look at connectivity. Where are these streams connected to? Are they connected to lake, ponds, or other rivers that are stocked? And we really do a good amount of background research and thinking and discussing before we choose a location. Once we choose a location, that's really where the fun comes in. So we go, like I said, we have what we kind of call missions or events. We usually try to have at least one in the spring and at least one in the fall. And what we do is that we go with our team. That can range from anywhere from 10 to 25 to 30 people. And we go out and designate certain areas that people go. And they go, and like I said, they fly fish. We always use barbless hooks. The difference between your normal fishing trip and what we do is once we catch the fish. So once we catch the fish, what we do in terms of taking a tissue sample from them is we clip a very, very small segment of the top part of the fish's tail. This is something, it's interesting. I've actually gone back to some of the streams that we fished before with friends and Trout Power members, and we've caught fish where we've actually seen, a tiny little piece of their tail taken out, but we can also see the regrowth that's happening. So when we started this, we talked to scientists who said, if you take a very small piece of the top of the tail, that will grow back. So it's nice to catch a few fish and see that regrowth happening and kind of get the confirmation that we're not leaving these fish, permanently affected by that. So what we do is, when we catch fish, we bag them, as we say. We carry these big gallon Ziploc bags. We immediately fill that bag with water and get the fish in the bag. So we're keeping these fish wet as the entire time that we're handling them, which is really important to us. We sterilize our scissors before we go into the field, and then we also do it every time we catch a fish. So we just take a butane lighter, take our scissors out, burn it for 5 seconds on each end, carefully remove that tissue sample, as I mentioned. We store our samples in the field in a 90% ethanol solution. We cap that vial. As soon as we get that fin clip and we get it within that vial, we immediately release the fish. So the fishes, again, we try to get them in and out as fast as we can. And then once we do that, we log where we are. So we have exact GPS locations connected to every single fin clip that we have, which is really important, especially when we map the genetics of these watersheds that we study. And then after we do that, we again re-sterilize. So we're at least sterilizing our scissors twice between every fish that we catch, because obviously contamination is certainly a concern of ours. So we want to do everything we can to ensure that we're preventing that from happening. And if we have any sample that we see that looks like that might have happened, especially when we have new volunteers who join us, we will kind of remove that fin clip from the set, just to ensure quality control. So this is the card that we give our volunteers, especially the first time they come and join us. In reality, this is probably a little bit more what it looks like. So catching a fish, there's it's my father up there on the top left with a nice smile and a beautiful wild brook trout. So catching the fish and bagging it, carefully clipping the fin, putting that in a vial of 90% ethanol, marking it on a GPS map, and then, like I said, making sure that fish swims away and returns back to exactly where we caught it. So that's our kind of our process within the field. So when people come and join us, and I certainly would invite anyone, I know it's a little bit of a drive to get up to the Adirondack Park, but it's beautiful in the springtime and just as beautiful, if not more beautiful in the fall, because there are no black flies. But if you ever come and join us for a mission, what you'll receive is a fin clip kit. There's an example up on the right side of the screen. Pair of scissors, lighter, and then usually 6 or 7 vials for those fin clips. We give all our volunteers a log sheet, so if they don't have a GPS device that can actually log their marking points, they can just take a paper form, and then when they come back, they turn in their fin clips, they turn in their log sheet, and then myself and a few others will go through and document that. And then each and every volunteer, we kind of tell them where to go, so we can really be intentional about what water we're covering, and we can really map what water we're covering. So in a 2 to 3 day fishing weekend, we can really expand and cover a lot of ground, which is fantastic. So, and then of course, when we're sending people out into the wild, in unknown territories, we're making sure that they feel safe, they feel comfortable, and that they're trained in any procedures that we have. And we've got a few doctors and safety officers on board, so we're always prepared and ready for the worst. But fortunately, I think the worst that we've ever had is maybe people having too many cocktails the night before on the fire, which is self-induced. There's no waiver for that. So, so once we do our field portion of our work, we come back, we bring our samples in, we hold them for the year. So if we have a mission in the spring and then a mission in the fall, at the end of that sampling season, towards the end of the calendar year, what we do is that we send them to a university for extraction and for analysis. For the past 2 years, we've been using Michigan State. And Michigan State, like I said, extracts the DNA samples and then sequences them. After they do that at 2 separate facilities, they send it back to Dr. Spencer Bruce. Dr. Bruce has been with us since the beginning of Trout Power. He works at the University of Albany currently, and he's the one who does all of our analysis and kind of reports back to us on what we found. So again, there's kind of that collaborative portion of citizen scientists, volunteer fishermen going into the field and collecting these fin clips, and then, sending them to universities, high-level institutions to continue our work for us. So here's just to kind of give you some examples of the sequencing that we do and then what we get back when we actually get this information back from Spencer at University of Albany. Up until 2020, we used a technique that was called microsat analysis. So microsat analysis at the time was a pretty new and pretty happening form of genetic analysis. And the way that this works is basically that we can go, and after the sequencing is done for a certain fish, Spencer gets this information back. Quick, and a lot of you may know this, quick high school science lesson here, that if you look at a sequence of DNA bases, there's really 4 letters, which is nice. There's A, T, C, and G, which are short for different nitrogenous bases. To make it simple, I just put 2 letters on this one, and G and C. But we get those sequences, and Spencer takes those, and what he does is he takes those sequences and we compare them to the 6 strains of brook trout that the DEC stocks within the Adirondack Park. So part of what we have to do is we have to run those samples, and then, like I said, we do a comparative analysis. So in this example that you see on the screen, again, really rough, generic example, we have, wild fish 1, wild fish 2, and then we've got our hatchery fish down below. So once we get those sequences back, we can do a comparison. So if we see a fish like we see in fish number 1, note how we kind of put the filter so it's nice and bright. But so fish number one, if we don't see any of those markers cross over, or very few of them, then we know that, okay, we've got a fish that's a native wild fish. We also see a lot of what we see in that second fish, which we see a mix of native genetics combined with hatchery genetics, which has been interesting and I dare say almost influential within our state, and I'll talk about why momentarily. The way that we actually get the data back and the way we display it, rather than using all of these bases of A, T, C, and Gs in these genetic markers, is that we are able to create basically bar graphs. So looking at the information in the center of the screen with all of those labels to kind of that more bar plot over on the right side, this is what we actually get back as a result. So it's a really wonderful visual analysis. It's great for presentations like this. It's great for us to look at when we get the results, because we're pretty much immediately just looking for a new color that's not one of our stock samples. So here's an example of that, and I'll show you examples from each year that we have. But up on the top of your screen, you have the 6 strains of brook trout. Using that microsat analysis, those are the 6 strains that are currently stocked within the Adirondack Park within New York State. So we have the domestic strain, the Temiskaming strain, the created domestic Temiskaming hybrid, and then the other 3 strains, the Windfall, the Little Tupper, and the Horn. Those are heritage strains of brook trout from stillwater bodies. So for example, Little Tupper is from a pretty well-known lake in the Adirondacks called Little Tupper Lake. And they've also started to stock those genetics within the park as well. So we compare the fish that we catch to those 6 strains. And then using the colors of those bar plots, we get results that you see like down on the bottom. So what we're looking for is we're looking for colors that are not one of the colors that you see at the top of the screen. So for example, down on this portion where you see this yellow coloration, or over here in this third set where you see this kind of salmon pink color, those are genetics that don't cross over with any of those stock strains. So to us, that shows uniqueness. That shows that biodiversity that we're really kind of out there looking to find and to show, you know, still is found in the Northeast and within the Adirondack Park. Now, that's what we did from 2016 to 2019. The genetic world, I've learned this through this organization, genetic world moves fast. And obviously in the past couple of years, it's had to really move fast. So in 2020, we decided to move to a new kind of sequencing, which is called RAD sequencing. Microsat, which was all the rage in 2016 when we started doing this, is quickly become not the rage, and I dare say almost outdated. So we made this switch also because we can learn a lot more about RAD sequencing beyond the genetics. We can look at family relationships, we can look at adaptation. So it's a change that we're hoping is going to really give us even more information than what we already have. So that's what we're working towards now. So quickly, just to kind of show you some results that we've had in terms of some of those bar plots. This is the first ever mission that Trout Power went on doing this genetic analysis. This was in a watershed that's in the central Adirondacks. If you've ever been to the Adirondacks, there's an area that's pretty well known to tourists called Old Forge. And this is a little bit north of the Old Forge area. So we did this, thought it'd be fun. Let's try this. Let's see how it goes. And as you can see on the right side, when we got it back and got these bar plots and compared them, we saw that blue coloration, and it was kind of like that holy crap moment. Whoa, look at that. You know, there's some unique fish here, which was pretty exciting for us, and definitely a shot in the arm into this whole idea that, you know, hey, we can do this, and, you know, let's see what's out there. So that's what we did in 2016. In 2017, what we did is that the area that we found and caught those fish was pretty far upstream in a watershed. And what we decided to do is move downstream, because downstream, it's connected to a lake, and that lake is stocked with brook trout by the DEC. So we actually went on either side of that lake to fish it. And what you can see in this example here is that for these samples collected in 2017, now when we compare our 2016 samples to the ones that we caught much closer to the lake, now comparatively, you can see a lot more of these stock strain colors coming in. that was really interesting to us, and also really interesting to the state. There was one person who was very adamant, sometimes relatively unkindly adamant, that wild fish and stocked fish do not hybridize, that you can put stocked fish of the same species over wild fish and they will not hybridize. So this data was an answer to that, and an answer that was pretty influential. And actually, and I take no joy in saying this, but actually that individual was so embarrassed by the claim and then the evidence we found over the given years that he actually stepped down from his position, which we certainly didn't want to happen, but kind of showed that if you're going to make a statement, I guess you'd better be very confident in it. So this was interesting to us, but then also interesting to the state, and had some influence, as we'll see momentarily. I mentioned the presence non-presence study that we did within the Silver Lake Wilderness over the spring of 2018 and spring of 2019 on 2 separate weekends. We fish at a number of different streams, as you saw on that map. And yet again, looking at the image to the left, I do know that these stock string colors, I know they vary a little bit, especially with the heritage ones. So sometimes they're purple, sometimes they're pink, but when you do the comparison from the left over to the right, again, we have this lovely sky blue color. So again, uniqueness. 2018 went to the upper headwaters of a larger river system called the Oswegatchie River. Didn't catch near as many fish, didn't sample as large an area, but still we see another example of, unique genetics. 2019, this is a project that was actually done in conjunction with the Trout Unlimited chapter. So we used our protocol, we trained Trout Unlimited volunteers who went to streams within their area for their chapter. And they funded the sampling. All we really did was train them and hook them up with our geneticist and our lab. And they found some extremely genetically unique fish, some of the most genetically unique fish that we found in all of our study. And one thing to me that was really exciting, and I think is worth mentioning, is that some of these fish, some of the cleanest fish genetically, the most pure fish, were in streams that were right next to roads. And I think that's awesome. I think that's awesome that rather than, because we've also had days where we've done 15 miles there and back just to hope that there are fish. So to have places that are in relatively populated areas that still have, pure native brook trout, that's pretty cool. That's pretty cool. So we're moving, we're grooving. We've had, years of finding and identifying these unique fish. And then obviously it seems like sometimes 5 minutes ago and sometimes it feels like 5 years ago, March 2020 hits. The world comes to a stop, and our world at Trout Power really comes to a stop in terms of our genetic analysis. We had to go about 2 years because of issues with not only supply chain, but just the large amount of, genetic work that was clearly being done around COVID, which I think is probably a priority over brook trout genetic analysis. So we're okay with it. But we had to stop. We had to stop. We were not allowed to do our genetic analysis. Last year, we were finally able to go back to having our large public events, which was so wonderful and a really great celebration. And we're finally able, we've got these materials, we've got labs now that are running results. So we're currently awaiting our results from those 3 years, and excited to see what they are, and really hoping that we can see similar results, because after looking at the ones we just did, one thing that I think is really cool for us is that every single year that we've gone out, every single one, 2016, 17, 18, and 19, and hopefully these 3 years as well, we've identified, a native brook trout with unique genetics. So it's kind of a nice hitting streak, if you will, if you're a sports fan, but it also is, again, just a testament to the wonderful biodiversity that still remains within the Adirondack Park. So the one other piece that we do is that we have one currently ongoing, and we're about to start another one, long-term monitoring studies. So one of the watersheds that we went and sampled, we found brook trout that were indeed had unique genetics, but also were brook trout that were greatly affected by acid rain back in the 70s, 80s, and 90s. And at times, there were people who we know who are involved in our organization and on our board and our members And they frequented that area and said that, you know, if you went there in the 90s and tried to catch fish, you wouldn't, you know, you never saw rises, you never saw fish. If you flipped over rocks, you know, you might see one caddis casing, but there were barely any macroinvertebrates, no frogs, no amphibian life, no salamanders. Now, if you go to that stream, caddis all over the place, huge green and brown drake hatches, and a really nice population of brook trout that we're seeing continue to rebound, not only in, you know, quantity of fish, but quality of fish. We are seeing the fish get bigger. So we thought, okay, well, we've got a unique population and clearly a pretty resilient population. As we know, I was talking to someone when we got on, you know, our weather has certainly been unique recently. Certainly our, you know, our world is changing, as it always has and as it always will. So we thought, well, you know what we're going to do? We're going to watch this population. We're going to see how their conditions and how their habitat change. And then through rad sequencing, we're going to see how those fish change alongside of it and in conjunction with it. They clearly had to adapt to survive the acidification that they experienced decades ago. So we want to see what they do now as we see this change continue to happen. So we continue to do our fin clipping. We identified sections of this watershed where we need to collect at least 20 fish or 20 fin clips per each year. We take water samples at our base flow chemistry as well as runoff and spring, trying to see groundwater influence, trying to see buffering capacity, looking at that water chemistry for various reasons. And then also throughout the watershed, we have temperature loggers both in the water and in the stream, as well as air temperature loggers at various locations as well. So we're, again, trying to monitor the habitat of these fish, as well as, you know, the fish themselves. The way that we do this in terms of the temperature loggers, we use something called onset hobo loggers. So over on the kind of the right center of your screen, that's one. They're very small loggers. You can see that in the palm of my hand. And then we build, just using PVC piping, build these kind of houses or casings for them. You can see that on the left. We take really, really large, long pieces of rebar that we kind of specially design, and a buddy Mark of ours welds to fit right through the holes that we cut in that piping, and we stake those into the stream. Obviously, we get permits to do all of this and, you know, say what the locations are. And then we put those in right at the beginning of the year. And by that, I mean, you know, ice out, so around April. And then we take them out usually just before hunting season, and try to let them go as long as we can. So we're going to do this for at least 10 years. This is going to be year 3 coming up of this study. And what we're almost hoping for is we're almost hoping to have 2 major events happen. Those events could be drought years. They could be years of extraordinary precipitation. They could be years of really dramatically late or early ice out. Again, we're just trying to see, as the habitat changes, you know, what is happening with these brook trout. There are some amazing studies, one currently going on in Michigan State right now, where it's all looking at the thermal or temperature-based adaptation of these fish. And as temperature changes, how quickly and how do brook trout adjust to that? And as we know, brook trout and our cold water fish, you know, as our water gets warmer, they're going to have to adapt, as they always have for thousands of years. So we want to be able to see what's going on. So that's really our big goal with this long-term study. The reason that we chose this watershed specifically is that if you look at this chart, and pardon me, one last time here, I'm going to get kind of science teachery. This chart shows the number of alleles within various populations that we have within New York State. Now, when I use the word alleles is relating to different versions of a gene, right? So if you think about something as simple as eye color, right? We have brown eyes, we have blue eyes, we have green eyes. All of those are different versions of genes that we have for the expression of eye color, as an example. Now, this fish, when we compared it to all the other fish, and it was Spencer who did this and our, you know, genetic workers at University of Albany. When they did this comparison, they found that this fish had the most allelic frequency of any fish that they had data on. So basically, in terms of adapting to change, they have the most tools in their toolbox to use, whether it be through mutation, you know, or just passing on of genes from generation to generation. So that's why specifically we chose to look at this population, the combination of their history with acidification in the watershed, as well as that high allelic frequency. So we're curious to see what happens. You know, again, it's only year 3, so it's really difficult to make any sort of, I would say damn near impossible at this point to make any sort of conclusion. But as the years go on, it'll be very interesting to see what we discover from this study. And like I said, we have one ongoing, and I think we're planning on starting up another one this year in a different watershed with some unique brook trout also. So the last thing kind of, and I know I've mentioned this and foreshadowed this once or twice, is that, you know, one thing that kind of caught our eye and caught the state's eye is I showed you this image earlier from 2016 and 2017 in our results, and the idea that we've seen data really showing the hybridization of wild fish and stocked fish. The reason that became important is that, other than just we can now see at the genetic level that this is happening, is that in 2018, the Department of Environmental Conservation in New York started to redo their inland trout stream management plan. And what they did is, like any large management plan being redone, they had, you know, public meetings, public sessions, both in person, obviously in person, and then after COVID, virtually and in person. You know, hearing what people had to say about, you know, when you go trout fishing, what are you looking for? You know, what do you like to do? What kind of fish do you like to catch? And one thing that they noticed was that they had a lot of people saying that, you know, we like catching wild fish. We like catching any sort of trout, but if we had our way, you know, we'd try to go find those wild fish. And then with some of the genetic work that we've done and some of the data showing that, hey, you know, if you put stocked fish on top of wild fish, they could hybridize, and clearly they do in some areas. That was partially influential in the new designation that we have within New York State. So now, if you look at the management of streams and rivers in New York State, they are either managed as stocked trout streams or as wild trout streams, meaning that if a trout stream has wild trout in it, they are not going to stock over top of them. Rather, their plan and their management plan really shifts to habitat improvement. So, okay, there are wild fish here. How can we make this habitat ideal for that population to continue to thrive? In areas that don't find wild fish, they're stocking fish. And now, because they're not stocking fish in some of those wild areas, now they're doing more stocking. So they have different designations, including stocked quality, stocked premier, where instead of just doing one stock in the spring, they're doing multiple stocking events because that's, you know, they're running it now as a fully put and take fishery. So I think that that's one thing that, you know, we know DC has said, you know, what we identified and what we discovered was something that they greatly took into consideration when thinking about this and thinking about wild fish and stocked fish and how to manage them. So over the years of 2020 and 2021, I saw a lot around New York State, electroshocking teams from DC going out and sampling every single stream to see, okay, what's here? What kind of fish are here? Are they wild? Have we stocked this previously? And so now, like I said, every single stream has one of those two designations, either wild or stocked, which we're you know, pretty happy and I would dare say proud that we were a part of having that happen to help to, you know, continue to advocate for our wild fish. So that's kind of what I was foreshadowing a little bit earlier. So so that's what Trout Power does. You know, we've kind of found a unique space in the fisheries and science world. You know, we don't do a tremendous amount. We do one or two things, but we do them pretty well. And I don't think, at least to our knowledge, that there's a lot of other groups that do this yet. So, you know, that's pretty cool. That's cool to me as a science teacher. It's cool to me as a fisherman. It's cool to me as a brook trout enthusiast. But I always kind of end presentations by saying this is, I guess, your sappy part. But the coolest thing about this to me, in any position I've held in this group, and now, you know, as the president of it, which really just means I send the emails instead of the meetings, you know, to be honest, but, and I get the best fishing spots. That's the perk. But, you know, the most unique thing to me is that seeing the amount of passion and in our volunteers We have people who come from all over the place. We have people who come, I know my cousin's on this call right now. He comes from Boston. We have people come from New Jersey, New Hampshire, Pennsylvania, all over the Northeast. And sometimes we're going to areas that, we don't know much about. We've just researched them and think there might be wild fish. So they're coming on the hope and a prayer just to maybe catch a fish. What was it, 4 years ago, two guys, John and Jim from Jersey, they had a 3-day float on the West Branch of the Delaware. And they canceled it to come to one of our events, and we had never been there. It's like, John, I appreciate it, but the Delaware, like that's pretty cool down there. And, Lord, I hope we find fish now. And we did. But it just, the passion that these people have to spend their own money to come and volunteer for us, to just maybe catch a fish, is really, really inspiring. And again, over the years, we've been able to catch and find some just absolutely beautiful fish. One thing that's really cool about these genetically unique fish is that probably the most striking thing about them and their different genetics is that we see fish that look different from each other. We've caught one of my favorite areas. If you look in this bottom left corner, we have a watershed that we fish and a brook that we fish where the fish almost have like a blue, like a cobalt blue hue to them. And we found fish with purple hues and more greens on their side. And they're just absolutely beautiful. So it's really inspiring to see people go, put the time, put the miles, put the money, and the hard work in to go and try to catch these fish. And the cool thing is that, again, we don't know what we're going to find. So very often we're finding fish like this. And then once in a while, and these are the really good days, where we go to a small stream in the Adirondacks and you drop a streamer or a dry fly on top, and you catch something like that. And that's not one of those little small brook trout. That's a fish that was just over 20 inches, caught on a little olive woolly bugger, in a really, really small pool, in a stream in the Adirondacks. And that's a wild native brook trout. And so when you see something like that still exists and is living and surviving on its own, that's the stuff that really inspires us and gets us going. So it's really it's a cool thing to be a part of. So in that, if you have any interest in what we're doing, whether it's just following what we're doing, we have a buddy of mine, Ryan, who's part of our group. He's really active with our page on social media. If you're on social media, we have an Instagram, troutpower.org. We have a Facebook, which is just if you search Trout Power. We have a website, troutpower.org. And then recently we've just started this year a membership. We've had a lot of people ask us over the past couple of years, hey, I want to become a member. How do I become a paying member? And we've said, well, we don't have that, but now we do, which is pretty cool. So $30 to become a member. And we actually have a higher tier of membership that's a little bit more money. But if you'd like to follow along with what we do, please check us out on any of those formats. My email is on the Trout Power website. And we are always open to people who are passionate volunteers that want to come and join us. You don't have to, as long as you like to fly fish and you don't mind going out and maybe walking through the woods a little bit. We always are looking for volunteers. So if you'd ever have interest in joining us at one of our missions and one of our events, we'd love to have you. And we're going to hope to put out our dates for at least the first half of 2023. We're going to hope to do that within the next month. And, so like I said, follow us in any way. And if not, or regardless, I certainly appreciate everyone listening and following along. And I know it's for me, I always do these presentations in the winter and then I immediately say, man, I want to go fishing. And now I'm going to walk outside the building I work in. I'm at school because the internet's better than my house up here. And I'll walk outside and it's 20 degrees and snowy. And you're like, oh, that's right. The water's frozen. Okay. I'll fish later. But again, I appreciate you all so much and hope everyone's enjoying the winter and doing well down there. And again, thank you all so much for listening. And obviously any questions that people have, I certainly will do my very best to answer.
I have a question.
So thank you very much.
The thermographs, the temperature, things you put in the water to monitor, you make those yourself?
I'm sorry, say that again. The graphs?
the temperature to monitor the water temps. That you make those yourself with the rebar and.
Oh, gotcha. Okay. So when we do those temperature loggers,
Make the houses work.
Make the house.
yes, we do. Okay, pardon me. Yes. So when we put those loggers in, yes, we make those just out of PVC piping, and we make those housings ourselves. Yep. We're fortunate. We've got a couple engineers that are part of our board or part of our membership. So they handle that process.
Those are actually the same loggers that we use. Hey, Chris, I have a question for you. So, always thinking one step further and trying to do more with less, so to speak. Do you think, Trout Power could be as successful as you are without the really strong, link you said you had to the New York DEC? Like, do you think that makes it, doable for you? Or like, I guess where I'm going with this is, could this be, could Trout Power be expanded to other states? Or do you think it's just unique because of the relationship that you have with the state?
Yeah, that's a great question, Mike. So first off, I think that we, most of what we do is independent of the DEC. It's obviously helpful to have a positive relationship with them. You know, anytime we call them for information, they're more than willing to share it with us. And like I said, they've called upon us before, but most of what we do is independent of them. All of those genetic studies, other than the presence, non-presence one, we've done that on our own. So it certainly benefits us, but we absolutely can function without them. In terms of moving to other states, you know, it is something we've talked about. I think that right now we really focus on the Adirondacks, mainly because our volunteers and our members, a lot of us have some connection to the Adirondack in some form. And also just the huge amount of water that's out there that isn't stocked with the potential to have brook trout. And, you know, I know there's other states in the Northeast. I'm in Vermont. There are those areas, not nearly as many in Vermont. New Hampshire, and then obviously Maine is a whole nother ball of wax. So we're focused on New York and the Adirondacks now. But I see no reason that in time we could go, or why we could not go out into another area and go and sample it. So DC is great to have on board with us, but we operate really independently and then just collaborate on specific projects.
Cool. Do you pay Michigan State for their analysis, or
We
do they have?
we pay them for whatever they charge. So when we send it to the lab, they send us an invoice. So yes, we do. Spencer, who at University of Albany, he's the man. He actually does it free of charge. He just loves doing this. He's a geneticist and loves doing stuff with fish. But to do the extraction, the sequencing, yes, we do. And that's where, you know, when we're doing fundraising efforts, whether it's membership or fundraising drives, a vast majority of our money raised goes to that sequencing, because depending on, in our past at least, to do one fish has ranged anywhere from about $55 upwards to, back in 2016, up to $100 per fish. So it is expensive to do that genetic work.
It answers my question. I was going to say, how much do you have to spend for the testing, but that's a pretty direct number.
It is. And like I said, that is when we look at our budget every year, that is where we allocate the most money by far, is that genetic analysis. So for the last 2 years, our budget looked, our bank account looked pretty good because we weren't able to do any of it. So, but then we sent it off and got the invoice, and it was like, well, never mind. There it goes. Now it looks like it normally does. But so yes, that is it. Definitely the largest expense that we encounter is that genetic work.
So Chris, another one for me, when people come to one of your events, because you didn't really go, unless I missed it, you didn't really go over it. So when people come to your events, is it a camping situation? Is it do you try to find lodging and everybody goes to the same place? Do you bring your own food? Like, how much do you guys do versus just, this is where we're going to be, you're on your own type of thing?
Yeah, great question. So I'll all of the above. All of the above. We have had events that range from actually staying at lodges. We've actually had the pleasure of staying at a couple Adirondack Great Camps, which was really fantastic. Obviously, that we had to pay for that, and anyone who wanted to join had to pay for their weekend in terms of staying at the lodge. We didn't make any money off of it, but the lodge did. But a lot of our events, and I would say a majority of our events, are camping based. So we find a location, we find either a campground nearby or just an area, especially primitive campgrounds we really like. And a lot of them are camping events. But in terms of bringing food and things like that, we always try to coordinate amongst our Trout Power members and board members to make sure that we're providing meals for all our volunteers. Obviously, you know, people bring snacks and, you know, bring their own beverages, whatever you enjoy drinking around a campfire. But we do try to make it as, you know, organized and comfortable as it can be when, you know, you're kind of organizing, you know, way out in the woods and very often outside of cell phone service. So we've done it from, like I said, from primitive campsites that you have to, you know, go down a narrow dirt road to get to staying at amazing, beautiful Adirondack great camps. So we have a variety of options when we do our events. I think knowing what we're going to do this year, I think our events this year are going to be primarily camping based.
Anybody else have any more questions for Chris? So Chris, I'm going to say thank you very much for still being at school this late at night. It's already and it was a great presentation. Please send me the information that I can share with the chapter.
Yes.
I know you had said that you thought the springtime one was going to be the first weekend in June.
I think so.
We have a regional here in Connecticut for TU that weekend, so I won't be coming to that one, but if you do one in the fall, I will be there.
Cool. That sounds good. Yeah, we are, I can say with the utmost confidence that we'll be having something in the fall, and we'd love to see you there, Mike, and anyone else who'd like to hop in the car and join. Like I said, always looking to you know, have volunteers and meet great people. That's the other thing that I fail to say in these presentations, but I've met some amazing, outstanding fishermen, advocates, environmentalists, and that's another true pleasure of this. So thank you all so much. Appreciate your time very much.
Thank you.
Thanks, everyone. Take care.
All pretty interesting. Cool. Any other questions or anything?
I think.
I'll just go ahead and end the meeting then. Thanks all. Chris.
Thanks, Chris.
Thanks, guys. See y'all. Take care.
Very much.